Hello dear students, this is Dr. Adwet and welcome to lecture number 10 of Cell, the Unit of Life. So, as far as this chapter is concerned, Cell, the Unit of Life, we have finished Introduction to Cell in lecture number 1. In lecture number 2, we spoke about history of microscopy and cell theory. In lecture number 3, we spoke about parts of cells, basics of genetics and characteristics of a cell. In lecture number 4, we spoke about the nucleus. In lecture number 5, we spoke about the various types of plastids. In lecture number 6, we spoke about mitochondria. In lecture number 7, we spoke about ribosomes and endoplasmic reticulum. In lecture number 8, we spoke about Golgi bodies, lysosomes, vacuoles. In lecture number 9, we spoke about cytoskeleton. And today, dear students, is lecture number 10. So if you have missed any of these lectures that you see here, I would strongly suggest that you please go through this entire playlist. You must be having the record of this with you as to how do you access these files. Please go through all these files and see the videos if required, and if you believe that you are not up to speed with any of them. Only once you are sure that you know all of these topics should you be watching today's lecture. So, as far as a eukaryotic cell is concerned, we have seen that a eukaryotic cell has cell wall and protoplasm. Protoplasm further comprises of the nucleus and its cytoplasm with the cell organelles. As far as the cell organelles are concerned, we have finished plastids, mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles and cytoskeleton. So all of these things are already done, which brings us to the next part of the eukaryotic cell, the last part, which is going to be the plasma membrane. So dear students, today's entire lecture is going to be on the plasma membrane. So today's lecture starts now. Today I will not have the opportunity to revise everything that we have done so far. It is not possible to revise 9 lectures in a matter of minutes, and that is why I insisted that if you do not have an idea or if you are slightly doubtful about any of the things that we've done in the previous 9 lectures, please have a look at it again, and only then should you be starting today's lecture. So as I said, today's lecture is about the cell membrane, the plasma membrane, also called as the plasma lemma. So if you were to see an animal cell or a plant cell, this outermost bluish whitish thing that you see here, the outermost bluish whitish thing that you see here is the plasma membrane, and that is what we are going to talk about today. Now, before we discuss the plasma membrane, it is very important that all of us are up to speed or at the same level as far as a few basics of chemistry are concerned. So dear students, the next 20 minutes I am going to talk about a few basic things in chemistry, and once we all are at the same level, up to speed in that bit, will you be able to understand the plasma membrane. So we will start from the absolute basics and we will work our way towards the plasma membrane. Let's begin. So as you can see on your screen right now, this is a carbon atom. Carbon has the atomic number 6, and what that basically means is that carbon has 6 positively charged protons in its nucleus, and it has 6 negatively charged electrons moving about 2 orbitals in a circular or an elliptical fashion surrounding its nucleus. So there are 2 electron clouds which are surrounding the nucleus, and the first one has 2 electrons, the second one has 4 electrons. So in all, as you can see in this carbon atom, there are 6 electrons which are going to be present. So an atom by definition has an equal number of protons and electrons. In the case of this carbon atom which you see here, it is going to be 6. So therefore an atom does not have any net electrical charge. This point is extremely important. Atoms are electrically neutral. They do not have a charge. And I just explained to you why they don't have a charge, because they have equal number of positively charged protons and negatively charged electrons, which will cancel each other out. Okay, moving on. However, atoms can gain electrons or they can lose electrons. So an atom can gain electron or an atom can lose electron. If it gains electron, of course it will become negatively charged because it will have more electrons as compared to protons. And if it loses electrons, then it will become positively charged because it will have more protons as compared to electrons. So an atom can get electrically charged, which means that it can become positively charged or negatively charged. Such charged atoms, which can either be positive or negative, are called ions. So this basic chemistry, I'm sure everybody knows, but I just wanted to revise that an ion is nothing but an atom which is electrically charged. It can either be positively charged or negatively charged. If it is positively charged, it will be called as a cation, and if it is negatively charged, it will be called as an anion. So you have atoms which are electrically neutral. Charged atoms are called ions, and they can be positive, called cations, or they can be negative, called anions. Okay, let's proceed. Electrons, as you saw in the first carbon atom, will be in shells or orbitals which are surrounding the nucleus. And each element, each atom in its configuration around the nucleus wants about 2 electrons in the first orbital, 8 electrons in the second orbital, and 8 electrons in the third orbital. Of course, depending on the number of electrons, there will be more orbitals, but for all atoms that are biologically important, इतना काफ़ी है कि फर्स्ट ऑर्बिटल में 2 चाहिए, सेकेंड ऑर्बिटल में 8 चाहिए, थर्ड ऑर्बिटल में भी 8 चाहिए. So to achieve maximum stability, this is what an atom wants. So atoms usually want this configuration in their outermost electron orbit. Hence, the atoms can either exchange or they can share their electrons. So if an atom does not, so let's say that an atom is going to have 11 electrons, which means that in its outermost orbital, it is going to have 1 electron. Now, it does not want 1 electron, it wants 8. So either it needs to gain 7 electrons to make it 8, or it needs to give away this electron to make it 8. So they will either exchange or they will share electrons. Such electrons which get shared or exchanged are called valence electrons. Again, basic chemistry, you must be doing this from 8th or 9th standard. So valence electrons are those electrons which are involved in or are available for possible bond formation. That is the use of these valence electrons, that they can either be exchanged or they can be shared if atom wants to chemically bond to another atom. So this brings us to our next slide. So atoms can exchange or it can share electrons. Now if an atom is going to exchange electrons, अगर उसको bond बनाना है, and if it is going to exchange electrons, what will happen? One will have to give the electron and the other will be receiving electron. मतलब कि जो 2 एटम साथ में आएंगे उसमें से एक देगा और एक लेगा. सो अगर एक देगा तो बाय डेफिनेशन अगर वो दे रहा है इलेक्ट्रॉन तो उसके पास इलेक्ट्रॉन कम हो जाएगा तो वो पॉजिटिव हो जाएगा, और जो ले रहा है उसके पास इलेक्ट्रॉन एक्स्ट्रा हो जाएगा तो वो नेगेटिव हो जाएगा. सो द मिनट यू विल एक्सचेंज इलेक्ट्रॉन्स, ions will be produced. Those atoms will no longer remain atoms, they will become ions. So one of the atom will give and will become positively charged, and one will receive and will become negatively charged. So the one which gives, of course, जो positive बन गया will be called as the cation, और जो negative है will be called as the anion. इस वजह से this kind of bond where the electrons are going to get exchanged is going to be called as ionic bonding. Ionic bonding. And now you can understand why this is called as ionic bonding. And in ionic bonding, subunits are always with charge. Subunits will always have charge. So let me give you an example here. So on the screen right now, here on this side, you are going to be noticing that there is a sodium atom and a chlorine atom. Sodium atom has the atomic number 11, which means it has 2 electrons in its first orbital, it has 8 electrons in its second orbital, and it has 1 electron in its outermost third orbital. So it will basically give this electron away to chlorine. Chlorine, which has 17 as its atomic number. So what is chlorine going to do? Chlorine will accept this and it will be able to complete 8 electrons in its outermost orbit. So sodium will give an electron and become a cation, positive, and chlorine will receive the electron and will become negative or anion. So in this kind of bonding, of course sodium will give it and chlorine will receive it, and as you can see in the diagram on that side, it will form something called as sodium chloride. And in sodium chloride you don't have sodium atom and chlorine atom, but you have sodium ion and chloride ion. So this, dear students, was a ionic bond. So I hope ionic bonding is now clear to everybody, and it is also clear to everybody that by definition those compounds which indulge in ionic bonding are going to have the formation of ions, and their subunits are going to be with charge. So that brings us to the next type of chemical bonding, where atoms which are coming together do not exchange electrons but share electrons. And you know this, dear students, this is going to be called as covalent bonding. Now the interesting thing is, in covalent bonding, the subunits may or may not have charge. The subunits may or may not have charge. And let me explain this a little bit in detail. If you were to look at this molecule here, so you can see that there are 4 hydrogen atoms on this side of your screen, and next to it there is going to be a carbon atom. As we all saw initially, carbon atom is going to have 6 electrons, which means that in its outermost orbit, instead of having 8 electrons, it is going to have 4 electrons. The hydrogen atom, on the other hand, is only going to have 1 electron in its outermost shell, which is also its first shell. So hydrogen will share electrons with carbon, carbon will share its electron with hydrogen, and together they will form methane, where in the outermost orbital their electrons are being shared, CH4, as you can see in the diagram on that side. So this methane is an example of a covalently bonded molecule. Now the interesting thing to know here is that in the case of methane, have a look at methane in this molecule, you will notice that electrons on this side are 3 in number, whereas electrons here are also 3 in number. So opposite sides pe electron ka number is the same. The distribution is even. If you see here, you have 2 electrons, and here also you have 2 electrons. So remember that in this case, where the electrons are being shared, in the example of methane, this kind of covalent bonding has equal distribution of electrons, or I can say even distribution of electrons. So अगर हम लोग covalent bonding की बात करें, we will notice that in covalent bonding, electrons are equally distributed in some covalent compounds, or they may not be equally distributed, and I will give you an example of this. But first let's talk about if electrons are equally distributed. In that case, the charges will balance out. There is equal distribution, even distribution, so charges will balance out, and there is no net electrical charge. There is no net electrical charge. Another example of this would be carbon dioxide. So you can see the carbon dioxide molecule here on the screen, and what you will notice is that the central black part is carbon and these 2 red molecules are oxygen. So however many electrons are on this side, a similar number of electrons are also going to be there on this side. So there is equal distribution or even distribution of charges. So this, dear students, was one type of covalent bond where electrons are evenly distributed. Now let's have a look at the other type. So when we see the other type, what can happen is that there is no equal distribution. So one or more atoms in the bond will hold the other electrons. मतलब कि इलेक्ट्रॉन्स हड़पने की ट्राई करेगा, उसके पास एक साइड पे ज़्यादा इलेक्ट्रॉन्स रखने की ट्राई करेगा. So in this case, the electrons will tend to cluster on one side of the molecule more than on the other side. So what will happen is that one side of the molecule will be slightly positive, and one side which has more electrons on that side clustered together will be slightly negative. So let me give you an example of this. An example of this is water, which you will now see on your screen. So notice in water, in this diagram that you see here, the red part is the oxygen atom and the blue part are the hydrogen atoms. So you see 2H and 1O, that's H2O. Notice that here in this part, you are going to have about 2 electrons. Here you will have about 2 electrons, but here clustered on this side are going to be about 4 electrons. So because electrons in a water molecule cluster on this side, it is shown as delta negative, meaning slightly negative. Whereas here, this part and this part are relatively slightly positive because they have fewer electrons. So if you compare this water molecule, to this carbon dioxide molecule here, you can clearly see that in the case of the water molecule, the distribution of electrons is not equal. So there is a specific charge on a water molecule. It is not electrically neutral. So wherever you are going to see the clustering of electrons, that part has charge, negative charge, slight negative charge, whereas the opposite side has slight positive charge. And this, dear students, is the reason why we say that water is a polar molecule. Polar is a word which is used for anything which has charge or direction. So we can say that water is a polar molecule because it is going to have charges because of uneven distribution of electrons. By the same principle or by the same logic, we can say that carbon dioxide, methane, they are going to be non-polar. They are non-polar because they do not have a charge or any kind of direction. So I hope that this particular chart is now clear to you that covalent compounds can either be polar or non-polar, and what does that mean? Polar covalent compounds are going to have charge, non-polar covalent compounds do not have any charge, the subunits do not have charge. So that brings us to this diagram, which is again the same water molecule, but you can see it in higher resolution here. So again you will notice that on one side there is going to be clustering of electrons, so it will be called delta negative. On the other side there is less clustering of electrons, or there are fewer electrons, so it will be called delta positive. So this is basically how the water molecule looks like. So dear students, now the important thing as far as biology is concerned for you people is this, that जो भी bonding की बात होती है will mostly either be ionic bonding or covalent bonding. Ionic bonding or covalent bonding. Covalent bonding is, as we just saw, further of two types. It can either be non-polar or polar. So this much now is very clear with everybody. The important point from a biological perspective is this, that ionic compounds or ionically bonded compounds have subunits with charge. The covalent compounds, which are polar, will also have subunits with charge. But the covalently bonded compounds, which are going to be non-polar, have subunits without charge, which means that they are electrically neutral. There is nothing positive or negative inside, they are neutral. So ionic cheezein are going to have charges, polar covalent cheezein are going to have charges, non-polar cheezein are not going to have charges. So methane, fats, oils, carbon dioxide, all these examples which we saw are basically going to be non-polar without any net electrical charge. Clear? Okay, fantastic. So with that in mind, now we can talk about this. Polar molecules have one side of the molecules which is slightly positive and one side of the molecule which is slightly negative. What this basically means is that polar molecules will be attracted to each other because they have slightly positive and slightly negative sides. So what does that mean? एक मॉलिक्यूल का जो स्लाइटली पॉजिटिव साइड है वो दूसरे मॉलिक्यूल के स्लाइटली नेगेटिव साइड को अट्रैक्ट करेगा. सो हर एक पोलर मॉलिक्यूल आजू बाजू के दूसरे पोलर मॉलिक्यूल को अट्रैक्ट करेगा क्योंकि उसके पास पॉजिटिव नेगेटिव साइड या कुछ तो चार्ज होगा जो ऑपोजिट टाइप के चार्ज को अट्रैक्ट करेगा. सो पोलर मॉलिक्यूल्स विल बी अट्रैक्टेड टू ईच अदर. So the slightly more positive part will be attracted towards the slightly more negative part, which means that there is weak bonding between the different polar molecules, polar or for that matter even ionic molecules in a polar solution. They are going to have weak bonds which are going to be formed, and these, dear children, you have to remember, are called hydrogen bonds. The definition of hydrogen bonds itself is the weak bonds which are going to take place between polar molecules or molecules who have subunits which have charge. So now you can understand that if we take water molecules, which you can see in the slide here, so you can see that there are about 5 water molecules, and if you will take the top one, you will see that this is its side which is slightly negative because of the clustering of the electrons, and uske niche jo molecule hai, you will see that this is the side which is slightly positive in that case. So they are going to get attracted towards each other, and as they get attracted, they will form hydrogen bonds. So in this diagram, the dotted lines that you see are showing you the hydrogen bonds, and you can just pause the video here and check all the dotted lines, and you will see that all the dotted lines are going to move towards the positive side to the negative side, from the positive side to the negative side. So this, dear students, what you see is the weak hydrogen bonds which are forming between the various water molecules in a solution. So now let's proceed. Because of the charges, most ionic and polar molecules easily dissolve in water. So now comes the most important point. So if you were to take a beaker, जरा उस corner में देखो, वो side में corner में, you will see that there is a beaker, and inside the beaker you will notice where I have shown the blue box, there is a cube of salt. Salt is sodium chloride. Now sodium chloride, as we just saw, is a ionically bonded compound. So in the case of sodium chloride, you will have Na plus, you will have Na plus, and you will have Cl minus. You are going to have Na plus and you are going to have Cl minus. Charges, charged subunits. Water, the whole molecule, some part will have a positive charge and some part will have a negative charge. So the minute you are going to put salt into water, the salt particles will break into Na plus Cl minus. Water molecules are already polar, so they will have positive and negative side, and all of this will start getting attracted to each other. And that is why salt is going to dissolve in water. And that, dear students, is why we say that these molecules who have subunits with charge easily dissolve in water. And that is why they are called hydrophilic. That is why they are called hydrophilic. So I hope that this slide is now clear to you as to why is it called as hydrophilic. Okay, moving on. By this same logic, non-polar substances don't form hydrogen bonds with polar molecules. So if you take any non-polar molecule which is electrically neutral and you put it inside water, which is going to have water molecules with, very small charges, they will not get attracted to each other. So they will not dissolve only, which means that non-polar molecules do not easily dissolve in water. And that, dear students, is why non-polar molecules are called hydrophobic. They are called hydrophobic. So ionic and polar are water loving, they dissolve in water. Non-polar do not dissolve in water and they are called hydrophobic. So you have hydrophilic and you have hydrophobic. I hope that this concept is now absolutely clear to all of you. If you have the slightest doubt about this, please just rewind this video by about 10 minutes and watch the whole thing again. But there should be no doubt in your mind moving forward as to what is hydrophilic and what is hydrophobic. Polar and ionic are hydrophilic, non-polar are hydrophobic. Is this absolutely clear with everybody? Okay, in that case, let's move forward. Now, in some large molecules, so there are some large molecules where it is not possible for the whole molecule to be having one affinity towards water. मतलब पूरा molecule जरूरी नहीं है कि water soluble होगा या hydrophilic होगा या पूरा का पूरा hydrophobic होगा, because this molecule can be made up of many subunits. So in some large molecules, what can happen is some regions are hydrophilic and some regions are hydrophobic. So to give you an example of a large molecule like this, we have something called phospholipids. As the name suggests, these are lipids which will have some phosphate moiety or will have some phosphate component to it. So they are called phospholipids. But a few of their components are going to be non-polar, fatty, do not dissolve in water, hydrophobic, and a few of their subunits are going to be polar, which will easily dissolve in water. So this phospholipid molecule that we are talking about is going to be something which is a large molecule having some part which is hydrophilic and some part which is hydrophobic. Such compounds, dear students, are called amphiphatic. So amphiphatic basically means that it is going to be both hydrophilic and hydrophobic at the same time. So if I was to just show you how this looks, this is how a phospholipid looks like. It appears to have a head with two fatty acid tails, So if we were to just simplify this diagram, it looks like this. that there is a polar or a hydrophilic head and you are going to have non-polar or hydrophobic tails. So I guess that this structure is now clear to you, that this is a simplified version of a phospholipid. A phospholipid which is going to have a polar head and it is going to have hydrophobic or non-polar tails. Amphipathic molecule. Is this now clear to everybody? Are you sure? Because the next concept is extremely important. So if you've understood everything so far, you will understand that why I started from atom. So I told you what is an atom, what is an ion, what is cation, what is anion, what is ionic bonding, covalent bonding, what is polar, what is non-polar, किधर charges है, किधर charges नहीं है, क्या hydrophilic है, क्या hydrophobic है. All of this I explained to you so that we reach this point that phospholipids are amphipathic with a polar water-loving hydrophilic head and two non-polar fatty acids से बने हुए hydrophobic tails. So all of that previous knowledge was to understand this. And now we come to the most important point. If we were to take a cluster of phospholipids, matlab a few thousand such molecules, I'm not talking one or two, I'm talking thousands of such molecules, and if we were to just throw them in water, what is going to happen to all these molecules? All of these molecules, once they hit water, this part which you see here is water loving and this part which you see here is water hating. So जैसे ही तुमने ऐसे हजारों molecules water में डाले, ये अपने आप को खुद से ही arrange करेंगे. They will arrange themselves on their own in such a manner that when they are dissolved in water, their heads will be towards water and their tails will be inside. They will spontaneously arrange themselves in such a manner that their heads will be outside and the tails will be inside. Why? Because the tails do not like water, so they are inside. And what will they keep on the outside? They will put all the hydrophilic heads which can touch the water because they are water loving. So the important points, dear student, is this, that when placed into water, phospholipids can spontaneously arrange into bilayers. Now what is the meaning of this? This is how they will arrange. So if you throw thousands of them in water, this is what the arrangement will be. Notice that ये side पे water है, ये side पे भी water है. So जिधर water है, बाहर सब जगह पे जिधर water है, what has happened? The heads have arranged themselves and उनकी tails अंदर की तरफ है. ये वाले भी heads बाहर water की तरफ है जो यहाँ पे है, and उनकी tails अंदर की तरफ है. So what has happened here, dear students, is the hydrophilic heads are facing outwards towards water, whereas the hydrophobic tails, the non-polar tails, they are inwards. So this, dear student, is a phospholipid bilayer. And now comes the most important point. Plasma membrane is a phospholipid bilayer. प्लाज्मा मेम्ब्रेन, क्लोरोप्लास्ट की बाहर की मेम्ब्रेन, क्लोरोप्लास्ट के अंदर की मेम्ब्रेन, माइटोकॉन्ड्रिया के बाहर की मेम्ब्रेन, माइटोकॉन्ड्रिया के अंदर की मेम्ब्रेन, सेल के अंदर के वेसिकल की मेम्ब्रेन, न्यूक्लियस की दोनों मेम्ब्रेन, सारी ऑर्गेनिक मेम्ब्रेन्स आर फॉस्फोलिपिड बाइलेयर्स. सारी ऑर्गेनिक मेम्ब्रेन्स आर फॉस्फोलिपिड बाइलेयर्स. So those of you who are still slightly confused, where has this word bilayer come from? Let me just show you one more diagram. So have a look at this diagram and see how this diagram represents a cell. This represents a cell. Can you see this? So this is the membrane of the cell. And notice कि ये cell के अंदर है और ये cell के बाहर है. So this will be called intracellular and this will be called extracellular. Inside the cell you have cytoplasm which is watery. Outside the cell also you have fluid medium. Notice बच्चों that everywhere on the outside you can only see the heads. You cannot see the tails. Even on the inside you are only seeing the heads which are water loving. And in between, sandwiched in between, you are seeing these yellow things which are nothing but those tails that we were talking about, the non-polar hydrophobic tails. So ये जो box तुमको दिख रहा है, ये box जो तुमको यहाँ पे दिख रहा है, can you see कि this box has been magnified here? So again, notice कि this is outside and this is inside. So here you can see is one layer which is made up of phospholipids. Can you see कि ऊपर की तरफ heads है और नीचे की तरफ उनके tails है? Can you see that? So this is one layer of phospholipids. You can also see that here is another layer of phospholipids. There is one more layer, but the difference is कि इनके heads नीचे की तरफ है और इनके tails ऊपर की तरफ है. But you have two layers. There is a layer on top and there is a layer below, and both these layers are going to arrange themselves like this. So what you have here is a phospholipid bilayer. So what you see here, dear students, is a phospholipid bilayer. अब I hope कि सबको ये समझ में आया कि ये लेयर ऐसे बना क्यों? Because ये साइड पे वाटर है, वो साइड पे भी वाटर है, तो जिस साइड पे वाटर है उस साइड पे हेड्स को रखे गए हैं और जो टेल्स हैं, जो हाइड्रोफोबिक नॉन पोलर टेल्स हैं, उनको अंदर बीच में सैंडविच किया गया है. So this, dear students, is what a plasma membrane looks like. And my entire lecture till this point was just to explain to you that plasma membrane is a phospholipid bilayer which has hydrophilic heads which are positioned outwards and hydrophobic non-polar tails which are going to be inwards. If this is absolutely clear to you, then we shall proceed forwards. But I think that if you've been paying attention so far, till now, it should be very clear to you that this is going to be a phospholipid bilayer. Okay, let's proceed. So this is how the phospholipid bilayer looks like. This, what you see here, is how the phospholipid bilayer looks like. And the interior region is hydrophobic. So बाहर की तरफ on top as well as below where you see the heads, it is water loving, but बीच वाला part is hydrophobic. And that, dear students, is the reason why polar and ionic molecules cannot pass. Polar and ionic molecules cannot pass from here, Now why they cannot pass from here? Because polar and ionic molecules have charge and this thing does not have any charge, so there is no attraction, so this will not allow it to pass at all. But there are some polar molecules like water and oxygen which are polar. Water is polar, oxygen is polar. It can move through this because of its small size. So remember, phospholipids, I told you in the beginning, is a large molecule having different subunits, having different polar and non-polar subunits. But water molecule is going to be very, very tiny and it can easily slip through between these gaps, the gaps that are there between the tails. So water molecules and oxygen can move in and out quite easily through a plasma membrane even though they are polar. But bigger polar and ionic molecules will not be able to pass. So keep this in mind before we continue further. So the interior region of the phospholipid membrane is hydrophobic. Polar and ionic molecules cannot pass, and some molecules like water and oxygen can slip through because of their small size. So that, dear students, brings us to the next part, which is movement of substances and molecules across the cell membrane. Movement of substances and molecules across the cell membrane. Now, right here we are going to discuss a few more basic things which will keep getting repeated in biology. So today itself I want you to focus absolutely clearly so that this concept becomes clear and you will not need any further revision. So the next few minutes, the next 10 minutes, what we are going to discuss is going to, as I said, going to be repeated again and again in biology. So I want all of you to focus on the next 10 to 15 minutes. So let's see how movement of substances and molecules happens across a cell membrane. So dear students, as all of you have done in your lower standards in school, in physics and chemistry, you know that things tend to go from higher points to lower points. And the reason for that is that the universe basically wants everything to be in equilibrium. So suppose if there is going to be something at a higher point and something at a lower point. The universe wants कि ये वाला इधर से इधर कुछ तो flow हो ताकि ये higher point नीचे आए और ये lower point ऊपर आए और ये दोनों equilibrium में पहुंचे. So what the universe basically wants is that things should go from higher to lower. So to give you various examples of this, you are going to have current which flows from higher potential to lower potential. You have wind which blows from higher pressure to lower pressure. And you have molecules which go from their higher concentration to their lower concentration. So to further elaborate on this point, let's talk about the last one, which is molecules moving from higher concentration to lower concentration. So let's say that I have a spray can, as you can see here, and if I was to spray it, the molecules of perfume that are inside or paint that are inside are going to come outside the can quite rapidly. So in the air surrounding the can, you are going to have a higher concentration of molecules, whereas the air in that far corner on the other side is going to have lower concentration of these red molecules. So what will happen now is these molecules will keep banging with each other in this area where they are of higher concentration, and they will slowly start moving towards the areas of lower concentration. So in this diagram, what is going to happen is they will move from higher concentration to lower concentration. So that's what's going to happen. They are going to spread out. This, dear students, is called diffusion. Movement of molecules from their area of higher concentration to their area of lower concentration is going to be called as diffusion. I'm sure you've done this concept in school. So what you see here is an example of diffusion. Molecules moving from an area of higher concentration to an area of lower concentration. Okay. At the same time, now I have the spray can at the opposite side, and we can repeat the same thing. The molecules will move from higher concentration to lower concentration. This again, dear students, is going to be called as diffusion. So why did I show you this can from two separate directions? Because I want you to understand that this is diffusion and this is also diffusion. So when you sprayed the paint or the perfume molecules from the upper can, they move towards their area of lower concentration, and even in the diagram below, given below, you can see the same thing, that the particles which were sprayed, the perfume particles or the paint particles, are moving from their higher concentration to their lower concentration. So in both cases, what has happened is diffusion. So that should now be crystal clear to you. With that in mind, we can come to the next point, which is the direction of diffusion is called gradient. The direction of diffusion is called gradient. So what is the difference, dear students, between diffusion and gradient? दोनों डायग्राम्स जो आपको यहाँ पे दिख रहे हैं उनमें डिफ्यूजन हुआ है, बट ऊपर वाला जो एग्जाम्पल है ये उसका ग्रेडियंट है. द ग्रेडियंट इज गोइंग फ्रॉम दिस साइड टू दिस साइड, एंड नीचे वाला है वो भी डिफ्यूजन है, बट उसका ग्रेडियंट उधर से ये साइड पे, उधर से ये साइड पे. So you can see in both cases diffusion has happened, but in both cases the direction was not the same. In the one which was above, it was going from here to here, whereas in the lower example, it is in the opposite direction. So दोनों में diffusion हो रहा है, but दोनों में gradient opposite है. So direction of diffusion is called gradient. So I hope with this example, you've understood the difference between what is diffusion and what is gradient. So you can say that gradient is the vector form of diffusion, the direction of diffusion. So once again, diffusion is the movement of molecules from their higher concentration to their lower concentration, and the direction in which it is going, right to left, up to down, down to up, that direction is going to be called as gradient. So gradient is always the molecules which are going from their higher concentration to their lower concentration. Okay, so I hope that this is now very clear to you. So let's take this concept a little further. So what I have here is a mountain, and I'm showing you a river which is flowing from the top of the mountain to the valley. Of course, if the river is flowing from the top to the bottom, that means that the water above has higher potential. So this is higher potential, this is lower potential. Of course, whenever things are going to go from higher to lower, that can be called as diffusion. And in this case, the direction is downwards, from up to down. So I can say, dear students, that this what you see here is the gradient. This what you see here is the gradient, up to down. Now, if I was to take one of you and I was to place you here on the top of the mountain and I was to ask you to jump into the river, even with zero effort, you do absolutely nothing, you will still reach the bottom without using any energy at all. You are still going to reach the bottom. Do all of you agree with that? Even if you don't swim, कुछ नहीं, खाली हाथ ऐसे रख दिए, all right, और खाली, of course you are just ensuring that you are breathing, but once you have done that, there is no effort required, you will easily reach the valley. So what happened here, dear students, is you traveled without using any energy. No energy was required on your part. So this activity, when you went along with the gradient, was completely passive, did not require any energy. So keep this in mind, that any process which is a downhill process, downhill matlab you are going from higher to lower. So if you are going from higher to lower, what is that direction of higher to lower? Gradient. So if you are going along the gradient, if you are going in the direction of higher to lower, you don't need to spend any energy and it will be a passive process. I hope this slide is clear to everybody, especially the things written in the top corner on that side, that along gradient is passive and it is also called downhill transport. Okay, so those of you who are now fully awake, let's see what happens when I ask you to jump here in the valley and I ask you that please swim along the river and meet me at the top of the mountain. So I want you to go in that direction. Dear students, I am now asking you to go from a lower potential to a higher potential. So mountain के ऊपर से नीचे अगर river आ रही है, तो that is going to be the process of things moving from higher to lower. That direction is going to be called as gradient, and in this case you are not going to swim along the gradient, but you are going to swim against the gradient. You are going to swim against the gradient. I think कि अब सबको ये समझ गया होगा कि तुम्हारी जान निकल जाएगी. It will require a phenomenal amount of effort to swim against the current, against the gradient. So as you go, you will reach. Undoubtedly, you will reach the top if you put in the effort, but it will require a lot of energy. So dear students, whenever you are going uphill, it is an extremely time consuming process and requires a lot of effort. So with this, the few basic things which I explained to you right now are these. As far as molecules are concerned, they move from their higher concentration to their lower concentration. This process by which it flows from higher concentration to lower concentration is called diffusion. The direction of diffusion is called as gradient. Whenever things go along the gradient, it does not require energy and is called passive. And whenever things go against the gradient, it requires a lot of energy and it is going to be called as active. This, dear students, should be crystal clear in your mind. So I want you to pause this video right now, take a screenshot of this and remember this. So please take a screenshot of this and write it in your notebook and remember it for future reference. That things always go from higher to lower. The process by which the molecules move from higher concentration to lower concentration is called as diffusion. The direction of diffusion is called gradient. Whenever things go along the gradient, it is called passive. Whenever things go against the gradient, it is going to be called as active. So if all of this is now clear to you, I can move on to the next part of today's lecture. But before that, let's quickly see what is the structure here. So here again, let's say that this is going to be a plasma membrane. Whatever is on this side will be inside the cell, whatever is on that side will be outside the cell. So this is intracellular, this is intracellular, and that is extracellular. Let's say that there is a non-polar molecule. गौर करें, I'm using the word non-polar molecule. Non-polar matlab not having any kind of charge. if there is a non-polar molecule on outside and it has higher concentration, let's say something like cholesterol or fat, and there is a lower concentration of that molecule inside the cell. In this case, it can easily move from its higher concentration to its lower concentration, because things always move from higher concentration to lower concentration, and in this case it will be able to move because it is a non-polar molecule, hydrophobic, water hating, and जो बीच वाला इधर लेयर है जो ग्रीन में दिख रहा है वो फॉस्फोलिपिड की टेल्स हैं, वो भी क्या है, हाइड्रोफोबिक, so they are also non-polar. So this is going to be called as diffusion, and non-polar molecules can easily pass. Now please understand this, dear students, that if this same thing was being spoken about with regards to polar molecules, कि पोलर मॉलिक्यूल्स का वो साइड पे हायर कॉन्सेंट्रेशन है सेल के बाहर और ये साइड पे कम कॉन्सेंट्रेशन है सेल के अंदर, तो ये डिफ्यूजन नहीं होता. क्यों ये डिफ्यूजन नहीं होता? Why this diffusion would have not taken place? Simply because polar molecules cannot pass. So it doesn't matter किधर ज़्यादा है किधर कम है, they will not go along the gradient because the polar and ionic molecules cannot pass through the hydrophobic non-polar tail वाला region, बीच का region. So anyways, as far as non-polar is concerned, I hope that this diagram is clear. So what about the polar molecules and the ionic molecules? What about them? How can they cross the membrane? So I think everybody now would be clear that you would require some road or some channel, या some bridge, some rasta through which they can go from outside to inside. So you will require some proteins जिनके बीच में ऐसा गली होगा, दिख रहा है? So you can see that this blue color का protein, which is actually here in this case a cylinder, and inside the cylinder you will notice that this is going to be a space or a lumen. So now if we are going to have some ionic molecule, it can pass through this. So this rightly, dear students, the blue thing which you see here will be called as channel protein. The blue thing which you see here will be called as channel protein because it's like a channel through which molecules can pass. So in this case, if you are going to have higher concentration on the outside and if you are going to have lower concentration on the inside of some ionic molecule, like let's say sodium, chloride, any kind of ionic molecule, it can go from its higher concentration to its lower concentration. It can go from its higher concentration to its lower concentration. Now please remember कि सोडियम, क्लोराइड, पोटेशियम, मैग्नीशियम सब कुछ ये ब्लू गल्ली में से नहीं आएगा. हर एक के लिए there will be a different channel protein because all of these are going to have different sizes. So if sodium molecule is small enough to pass through this lumen, this white area, then sodium molecule will pass. But a chlorine molecule, if it's very big, if a chlorine molecule is very big, it will not be able to pass. So the idea here is that channel proteins are highly specific. But let's say that this channel protein was for sodium and sodium बाहर ज़्यादा है और अंदर कम है, तो sodium will go from its higher concentration, to its lower concentration. What will we call this, dear students, when things go from higher to lower? Diffusion. But will this diffusion happen without this channel protein? Will it happen? No, because ionic molecules cannot pass unless there is a gully like this, unless there is going to be a channel protein like this. And that is why, dear students, this what I just explained to you is called facilitated diffusion. The blue color का channel protein is facilitating, मदद कर रहा है diffusion को होने के लिए. अगर वो blue protein नहीं होता तो ये higher to lower जाता ही नहीं. Higher to lower इसलिए जा रहा है because वो blue protein वहाँ पे है which is allowing it to go. So without the blue protein it did not matter कि किधर higher था किधर lower था, movement ही नहीं होता. So this, dear students, is what is required for polar and ionic molecules to cross the membrane. You require channel proteins. This kind of diffusion is called facilitated diffusion. This kind of diffusion is called facilitated diffusion. Okay. And finally, what if you have ionic molecules you want them to pass through the membrane, but they have to go from lower concentration to higher concentration. मतलब लेट्स से कि अंदर सोडियम बहुत ज़्यादा है, लेट्स से कि अंदर सोडियम बहुत कम है, and you want the sodium to go outside. So inside there is less sodium, outside there is more sodium, but the cell does not want sodium at all. So the cell is like बाहर फेंको सोडियम को, बाहर फेंको, even though अंदर कम है और बाहर ज़्यादा है. So now in this case, dear students, will the sodium molecules go from their higher concentration to lower concentration, or does the cell want to throw them out from lower concentration to higher concentration? So you are right, the cell wants to throw them from lower concentration to higher concentration. अब सोच समझ के जवाब देना, ये एग्जाम्पल में ग्रेडियंट किधर है? Gradient is always from higher to lower. So gradient is from outside to inside. But the cell wants to throw sodium from inside to outside. Not along gradient, but against gradient. And that, dear students, will require energy. And you know that cells use energy in the form of ATP. So that, dear students, will require ATP. Is that clear to you? Fantastic. And if it requires ATP, that blue color ka channel protein will now be called as a pump. Why will it be called as a pump? Because धक्का मार रहा है, एनर्जी लेके धक्का मार रहा है ना सोडियम को, जिधर कम है टू जिधर ज्यादा है, अगेंस्ट द ग्रेडियंट. दिस इज अपहिल, you are going against the gradient, so will require energy. So this, dear students, is not going to be diffusion, because diffusion is from higher concentration to lower concentration. That direction is called gradient. Here you are going against the gradient. So this kind of transport across a membrane has a special name. It's called active transport. It's called active transport. So I just explained to you that movement across a membrane can be simple diffusion, it can be facilitated diffusion, or it can be active transport. I hope these concepts are crystal clear in your mind. They are going to be repeated in multiple chapters in biology. So if you understood this today, the same thing is what I will be repeating in other future lectures as well. So keep all of this in mind. Alright, so here's what NCERT has to say about this. So now these are lines from NCERT which you will clearly understand. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can briefly cross the membrane without any requirement of energy. This is called passive transport, without requiring any energy. And this passive transport, ध्यान से सुनना अभी, ध्यान से सुनना, non-polar molecules के लिए easily passive transport होता है. Polar और ionic molecules के लिए भी होता है passive transport along gradient, but उसके लिए चाहिए proteins, channel proteins, तो ही होगा facilitated diffusion. Clear? Okay. So neutral solutes, जो अभी मैंने कहा neutral चीज़ है, non-polar, may move across the membrane by the process of simple diffusion along their concentration gradient from higher concentration to lower, जो पिछली 15-20 मिनट से हम लोग कर रहे हैं. Water, even though it is polar, blue वाला NCERT में नहीं दिया, मैंने आपके लिए लिखा है. Water, even though it is polar, may also move across this membrane from higher concentration to lower concentration because of its small size. As the polar molecules cannot pass through non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. So I hope कि अब ये वाला लाइन सबको समझ में आया कि पोलर या आयोनिक मॉलिक्यूल्स क्रॉस नहीं कर पाएंगे मेम्ब्रेन के नॉन पोलर वाले टेल वाले हिस्सों को. सो उनको चैनल प्रोटीन्स लगेगा विच विल अलाउ देम टू ट्रांसपोर्ट देमसेल्व्स. And this transport is called facilitated diffusion if it is from higher concentration to lower concentration, and it is called active transport if it is from lower concentration to higher concentration. Simple. So few ions or molecules are transported across the membrane against their gradient from lower to higher. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. The example given in NCERT is sodium potassium pump. This sodium potassium pump we will do in detail when we are going to do this in another chapter called as neural control and coordination. So ये अभी डिस्कस नहीं करेंगे, अभी सिर्फ याद रखो कि an example of a pump in NCERT is sodium potassium pump. Okay, fantastic. Moving on. So this is a beautiful diagram of a phospholipid bilayer, and I have split the membrane so that you can see that there are two layers, and that is why it is called as a bilayer. You can also see various other things in the membrane. For example, you can see the channel proteins that I am talking about. You can see some other proteins which have other functions which we will subsequently see. But there are many proteins in the membrane, there are cholesterol also, there are these chains of polysaccharides which are also attached. So बहुत सारी चीजें membrane में है. So what does the NCERT say about it? The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. So from previous lectures you know that electron microscope was required to see the details within the cell. So 1950s तक ऐसा दिखता है वो तो मालूम ही नहीं था. But 1950s तक we knew कि what was the composition, किन चीज़ों से membrane बना है. So we knew the chemistry of the membrane, and that was basically because of the RBCs, mammalian RBCs. So chemical studies on cell membrane, especially in human red blood cells, enabled scientists to deduce the possible structure of the plasma membrane. So 1950s में chemistry भी आगे बढ़ रहा था, microscopy भी आगे बढ़ रहा था. So माइक्रोस्कोपी में ऐसा दिखा, उसके पहले हमको केमिस्ट्री की वजह से पता था कि देयर आर गोइंग टू बी ए लॉट ऑफ दीज केमिकल स्ट्रक्चर्स विच आर देयर इन द प्लाज्मा मेम्ब्रेन, एंड मोस्ट ऑफ दिस वाज डन ऑन ह्यूमन आरबीसीज और आरबीसीज, मैमेलियन आरबीसीज. These studies showed that the cell membrane is composed mainly of lipids and proteins. So phospholipids वाला lipid part and proteins जैसा कि हमने देखा, channel proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head outwards towards the outer sides and the hydrophobic tails towards the inner part. That is what we have been discussing in the entire lecture today. So now this line should be very clear to you. This ensures that the non-polar tail of the saturated hydrocarbons, वो fatty acid वाला tail, is protected from the aqueous environment. In addition to phospholipid, membrane also contains cholesterol के molecules. Later, biochemical investigations clearly reveal that the cell membrane also possesses protein and carbohydrates. So phospholipid और cholesterols है, proteins and carbohydrates भी है. So all these things are together forming the phospholipid bilayer, plasma membrane. The ratio of protein and lipid varies considerably in different cell types. So plasma membrane हर एक सेल का ऐसा ही दिखेगा, बट उसमें कितने प्रोटीन्स है, कितने लिपिड्स है, कोलेस्ट्रॉल कितना है, विल वेरी फ्रॉम सेल टू सेल. लिवर के सेल में कितना होगा विल बी डिफरेंट देन आरबीसी में कितना होगा. सो द रेशियो ऑफ लिपिड्स एंड प्रोटीन्स वेरीज कंसिडरेबली इन डिफरेंट सेल टाइप्स. इन ह्यूमन बींग्स दे हैव गिवन इन्फॉर्मेशन फॉर आरबीसी. ह्यूमन बींग्स का सबका नहीं बताया, बट आरबीसीज या एरिथ्रोसाइट्स जिनके ऊपर सबसे ज्यादा रिसर्च हुआ है उनके बारे में NCERT has mentioned that 52% is protein and 40% are lipid. Of course, that is only 92%. The remaining 8% are going to be the polysaccharides, cholesterol, and other things like that. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. So now this is something I did not mention earlier, but it is very easy to understand. If there is a channel protein like this, ये दिख रहा है? अब ये पूरा का पूरा मेम्ब्रेन में घुसा हुआ है और आर पार जा रहा है. It is going through and through, which means that this will not be easily removed from the membrane, and that is why it can be said to be an integral protein. It is a part of the membrane itself, very difficult to remove, difficult to extract. But if you see some other molecules here, some other proteins here, they are just embedded and may be easily removed. So these ones here which can be easily removed are going to be called as peripheral. So peripheral protein lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. So depending on ease of extraction, you can have either integral proteins or peripheral proteins. Now, look at this animation on your screen right now, and you will notice that all these proteins which are in the membrane, you can see this beautiful animation on this side that all these proteins, especially have a look at this protein here, this pink color ka protein, ye dekho. Also have a look at this protein here, look at this protein. See how things are going from inside to outside and how all of them are always moving. All these proteins are continuously moving. They are not at one point. Because of it, because of this particular movement, it is said that the plasma membrane has fluidity. So this fluid motion of things continuously moving, it is not remaining in the same state. It is dynamic. It is changing continuously. Because of this, it is said that the membrane is fluid. So this was proposed in 1972 by two scientists, as you can see here, Singer and Nicholson. So an improved model of the structure of cell membrane was proposed by Singer and Nicholson 1972 and is widely accepted as fluid mosaic model. So ये mosaic क्या है? I will tell you in 2 minutes, but I hope that the fluid bit is clear to you. So according to this, the quasi-fluid nature, quasi matlab almost fluid nature where things are moving in and out and the proteins and the polysaccharides are moving all along the membrane. So the quasi nature of the lipid enables lateral movement of proteins within the overall bilayer. So please remember that these proteins which you saw, which were moving, they are moving side to side. Side to side is called lateral. They are not moving up and down. They are not moving inside outside. They are moving side to side. So the nature of the lipid enables the lateral movement of proteins within the overall bilayer. And this ability to move within the membrane is measured as its fluidity. The ability of the peripheral and integral proteins to move within the membrane, as you saw in the animation, is called as its fluidity. The fluid nature of the membrane is also important from the point of view of cell growth, formation of junctions between the cells, जो हम लोग आगे करेंगे, secretion, endocytosis, cell division. So you already know endocytosis. Cell division is an entire chapter that we are going to do further. But for all these things, it is important that the membrane has fluidity. Right? So lastly, I want you to see this picture. I think many of you must have seen this at some point in your life. These are small tiles of various colors. Literally tile, ladi ke tukde hain, and they have been put on a wall to create this beautiful design. So this beautiful design, which is made of little subunits, the tiles, the broken pieces of tiles of different color. This, dear students, is called mosaic. This is called as a mosaic. So this is the plasma membrane, and as I just explained to you, it has fluidity. It has fluidity. The proteins, the peripheral and the integral proteins, जो easily निकलते हैं, peripheral जो एकदम embedded है, integral, they keep moving in this membrane laterally side to side. That is called fluidity. At the same time, this membrane on its surface, if you look at this surface, it has design. You can see pink, green, yellow, pink, blue. You can see it like this, like design. And this design is called mosaic. And that is why this model of the plasma membrane, dear students, is called as the fluid mosaic model. 1972, Singer and Nicholson. And this is given as this diagram in the NCERT. So if you've been paying attention for this entire lecture, this should now be absolutely clear in your mind कि ये ऐसा membrane कैसे आया. So that, dear students, finishes off the entire protoplasm part of a eukaryotic cell. हमने nucleus देखा, हमने सारे cell organelles देखे, and आज एकदम detail में we finished everything related to the plasma membrane. So the only thing left about eukaryotic cell now is going to be its cell wall, and that cell wall is also not present in plant as well as animal cell. It is present only in the plant cell. So we will see the cell wall and its structure in the next lecture, along with the difference between a plant cell and an animal cell. And with that, in the next lecture, lecture number 11, we will finish everything related to the eukaryotic cell. So thank you, dear students. I hope you have understood everything that we have done today, and I cannot wait to see your smiling faces in the next lecture. Thank you. God bless. Work hard. Be nice.